All right. Hi, everyone. I'm going to present some work that I recently submitted, which is a collaboration with Stu West and Alan Graffin. So explaining altruism is fundamental to understanding how life evolved from simple replicators to complex animal societies. The problem of altruism is, why should an individual perform a behavior that's costly to itself, but benefits another individual? The most extreme form of altruism is altruistic sterility, where an individual gives up its reproduction entirely to help others to reproduce. So this includes social insect workers, stalk cells in bacterial and slime mold fruiting bodies, and fungal cells that sacrifice themselves to protect other cells from the host immune response. The standard explanation for altruism is provided by kin selection theory. So this shows how altruism can be favored if it's directed towards pedigree relatives, so siblings, etc., because those relatives are likely to carry the same altruistic gene. In recent years, an increasing number of examples have been proposed for an alternative route to altruism called the greenbeard effect. So greenbeard genes cause their bearers to produce a recognizable signal, such as a green beard, recognize that signal in others, and then behave altruistically towards signal bearers. And so by directing altruism towards individuals that share the same gene, greenbeards could favor altruism towards individuals that are not pedigree relatives, like siblings and so on. And suggested examples of greenbeards include a queen-killing gene in fire ants, toxin-antitoxin systems in bacteria, and allo-recognition genes, which control cell fusion in social bacteria and eukaryotes. However, it's not clear whether these examples really represent greenbeard adaptations. So it's possible that genes with greenbeard-like properties could be favored by kin selection between pedigree relatives, with their apparent greenbeard features arising incidentally rather than adaptively. For example, in populations with limited dispersal, it's possible that interacting individuals tend to be pedigree kin, meaning altruism would be favored even without the greenbeard-like signaling and recognition. Or perhaps individuals that share the greenbeard tend to be pedigree relatives, meaning that the greenbeard-like properties just serve as a mechanism to recognize kin. So given this, what counts as a greenbeard adaptation? So I suggest that a gene with the properties of a greenbeard, so signaling, signal detection, and altruism, should, in the first instance, be called a candidate greenbeard. And a candidate greenbeard only counts as a genuine greenbeard adaptation if it's maintained, at least in part, because these properties allow it to direct altruism towards identifiable copies of itself in other individuals, including in pedigree non-relatives. The alternative is that the gene is maintained because altruism is directed towards pedigree relatives, or because the gene also leads to some other direct benefit to its carrier. So to formalize this, I suggest that a candidate greenbeard must satisfy two conditions to qualify as a greenbeard adaptation. So first, an altruism condition. So if the costs and benefits of altruism are removed, the gene should no longer be maintained to the same extent. So this ensures that altruism itself contributes causally to the gene's evolutionary stability, rather than being an incidental byproduct of selection on some other trait. And second, a linkage condition. So if the signaling, recognition, and altruism are genetically unlinked, then at least one of these components should be destabilized. So this ensures that physical linkage forms part of the selective explanation for the system's maintenance. So if a candidate greenbeard passes both conditions, then this implies that the gene's maintained by the greenbeard effect and that it represents a greenbeard adaptation. Failing the altruism condition implies that the candidate greenbeard is maintained because it provides some non-altruistic direct fitness benefit, so it doesn't count as a greenbeard adaptation. And passing the altruism condition but failing the linkage condition implies that the candidate greenbeard is maintained by kin selection with the linkage incidental, and so again doesn't represent a greenbeard adaptation. So now that we've formalized greenbeard adaptations, we can use theory to ask how likely they are to arise. So I analyzed a multi-locus population genetic model, and I examined firstly when candidate greenbeard genes are stable, and secondly whether candidate greenbeard genes are destabilized by when altruism or linkage are removed. So I'm modeling a viscous population. I'm tracking evolution at two loci. So one locus determines whether a signal, so in other words, a beard, is produced, and also what color the beard is. And the second locus determines whether altruism is expressed conditional on matching beard color. And the reason why I have these as separate loci is that I can unlink them to test the linkage condition. I assume that individuals with beards attempt to form a social partnership by randomly encountering nearby individuals. And when they encounter an individual with a matching beard color, or rather without a matching beard color, they reject that partner with a probability alpha and initiate a new social encounter. And this process continues until either the individual finds a beard-matched partner or fails to obtain a partner at all. And so the parameter alpha captures the extent to which individuals can actively search for beard-matched social partners. I assume that individuals who find a beard-matched social partner have the opportunity to be altruistic. So altruism confers a benefit B on the recipient and imposes a cost C on the actor. I also allow there to be an additional pleiotropic benefit, D, which is linked to altruism. And so a biological example is the queen-killing supergene in fire ants, which encodes a range of things alongside the greenbeard traits, such as the number of queens in a colony, dispersal strategy, and so on, which have direct fitness consequences. And I also have random mating and meiosis in the model. And so analyzing it, I found that candidate greenbeards can be maintained in a few different ways. So sometimes only one type of signal is maintained. So these are single color candidate greenbeards. And single color candidate greenbeards can be maintained in two ways. The first is by high population viscosity. So in panel A, candidate greenbeards are stable above the gray line. So as population viscosity increases along the x-axis, the gene is more likely to be stable. However, the gene is stable even without linkage above the purple dashed line. And we can see that the gray and purple lines converge for high F. So although population viscosity makes it more likely that the candidate greenbeard is stable, it also makes it more likely that the linkage conditions failed. So candidate greenbeards maintained by population viscosity likely represent kin-selected rather than greenbeard adaptations. The second way that single color candidate greenbeards can be maintained is by linked direct benefits. So in panel B, as the linked direct benefit increases along the x-axis, the gene's more likely to be stable. However, the gene is stable even without altruism above the orange line. So although, and we can see again that the gray and orange lines converge for high D. So although linked benefits make it more likely that the candidate greenbeard is stable, they also make it more likely that the altruism conditions failed. So these genes are likely maintained for their linked private benefit rather than as greenbeard adaptations. And so overall, for single color candidate greenbeards, or rather for single color candidate greenbeards to represent genuine greenbeard adaptations, we need F and D to be high enough that the gene is stable, but not too high. Otherwise, the greenbeard effect is not required for its evolutionary stability. So in other words, we need a kind of Goldilocks combination of parameters, which we might not expect to see very often in nature. I also found that sometimes many types of signal are maintained at once. So these are multicolor candidate greenbeards. And I found that multicolor candidate greenbeards are maintained above the green line in the figure. So on the x-axis, we have the extent of linkage between the greenbeard traits. And we can see that this has no effect on whether multicolor candidate greenbeards are stable. So this means that multicolor candidate greenbeards always fail the linkage condition, meaning they never represent greenbeard adaptations. And instead, we can see from the y-axis that multicolor candidate greenbeards are only stable when there's a high social encounter rate, alpha. And this result agrees with recent work on the evolution of genetic kin recognition. So it's been shown that when altruism is conditional on matching a genetic signal, individuals bearing common signals are more likely to be recognized and helped, whereas individuals bearing rare signals are less likely to receive help. And as a result, common signals increase in frequency, which eliminates the signal diversity that's required for genetic kin recognition. And this problem, where genetic kin recognition can eliminate the genetic diversity that's needed for genetic kin recognition, is called Crozier's paradox. And it's been shown that multiple social encounters, so high alpha, can eliminate this problem by allowing individuals with rare signals to locate matching partners. And so we can see from the figure that I recover this result. So overall, then I find that the signaling and recognition parts of a multicolor candidate greenbeard just act as a mechanism to identify and preferentially help pedigree relatives. So multicolor candidate greenbeards are maintained by kin selection, not as greenbeard adaptations. And so now we can reevaluate suggested examples of greenbeards in light of the model. So starting with multicolor greenbeards, suggested examples include those controlling cell fusion and altruistic stalk formation in Dictyostelium slime mold and Myxococcus bacteria, specifically the TGR and the TRA genes, which are highly polymorphic. So my model predicts that the linkage conditions failed for these scenarios. So the genes therefore represent mechanisms for identifying closer pedigree relatives. So in other words, they're mechanisms for genetic kin recognition. They're unlikely to represent greenbeard adaptations. And consistent with this, in every case that's been examined to date, strong population viscosity ensures that cell fusion mediated by multicolored candidate greenbeards occurs almost exclusively among pedigree relatives. So moving on to single color greenbeards, two types have been suggested. One type is where there's also strong population viscosity, meaning the altruistic benefits go to pedigree relatives. So this includes the CSA gene in dictyostelium, FLO1 in Saccharomyces, and a range of toxin-antitoxin or bacteriocin systems in bacteria. So my model predicts that the linkage condition is failed in these scenarios, and so they represent kin-selected rather than greenbeard adaptations. And the other type of single-color greenbeard is where the gene also provides some other linked benefit to the carrier of that gene. So the key example is in fire ants, where workers carrying an allele selectively identify and kill queens that lack the allele, which secondarily benefits queens which have the allele. So this allele additionally controls both queen number and multiple life history variables, where the optimum appears to depend on the number of queens in the colony, such as size and dispersal strategy. And importantly, these direct benefits are likely to be sufficient to maintain the gene, even in the absence of any indirect benefits associated with queen killing. So I think that the gene therefore fails the altruism condition, meaning it's maintained for its direct fitness benefits rather than as a greenbeard adaptation. And finally, there are several examples which have been suggested as greenbeards, but which lack the properties to even be a candidate greenbeard. So this includes side-blotched lizards, which exhibit signaling, recognition, and altruism, but these traits are not physically linked. And so overall, there's no clear empirical evidence for greenbeard genes. And I'll just end with this quotation from George Williams, who's been mentioned a few times. So, and it's from his 1966 book, Adaptation and Natural Selection. So he said that adaptation is a special and onerous concept that should only be used where it's really necessary. In explaining adaptation, one should assume the adequacy of the simplest form of natural selection, unless the evidence clearly shows that this theory does not suffice. And so when Williams wrote this, he was criticizing explanations for adaptation that require a benefit to a group of individuals. And his argument is essentially that Occam's razor compels us to reject such explanations unless more parsimonious explanations can be shown to be insufficient, such as explanations that require a benefit only to individuals. And I think this same logic applies here. So greenbeard explanations for altruism require a very particular stipulation on genetic architecture, namely the linkage of three distinct traits. And I think we should only accept such an explanation for altruism if we can definitively rule out simpler explanations, for instance, those that require only a benefit to individuals rather than additional genetic details. And looking across the empirical examples, I don't think the standard's been met. And so there's maybe no reason to think that the greenbeard effect has played an appreciable role in the evolution of altruism. I'll leave it there. Thank you.